Okay. I'm the replacement of Maria Schumacher. I'm not Maria. So, and I'm not talking about her stuff. I'm talking about my research here. So I'm working on Mucor. So there's some basic science about mucodimorphism and how dimorphism affects virulence and how calcineurin regulates that aspect and how calcineurin can be targeted to treat mucoinfections. So mucormycosis is not really common fungal infections, but keep increasing. Then problem is really aggressive fungal infections. It's not one species responsible for this. There's a mucormycosis fungi responsible for this, including Mucorrhizome, Pursorhizome, Mucorcunningham, and several other more species. So infection mainly occurs in immunocompromised patients, and sometimes you can see so immunocompetent host can develop mucormycosis too. So mainly associated with the transplant recipients and also blood cancer patients. Unfortunately, if you have disseminated infections, so mortality is unacceptably high. So as I mentioned, diabetes, steroid, and other immunosuppressive conditions increase the chance of incidence, and high serum iron level increase the risk of the infections. However, so as I mentioned, zygomycosis or mucormycosis is increasing, but this is an incredibly understudied area. So actually mucormycosis hit the Joplin, Missouri, which is disrupted by tornado. So several patients actually died of mucormycosis after tornado devastation. So in a children's hospital in New Orleans, several kids developed mucormycosis. They finally died. So actually, hospital linen brings mucospores to the hospital. They develop mucormycosis. This is just kind of an example. So mucor is my favorite organism. So you know Robert Hooke, who invented the microscope more than 300 years ago. With his first microscope, he observed the little tiny things, then described and drew that thing in his book, Micrographia. In the chapter 20, you see this picture, and he described the vegetative body, long cylindrical, transparent stalks with the weight of round and light, and several of them can be broken like that. If we touch that, it broke things. So we have here a little bit better microscope than he had. So this is our image of mucor. Long cylindrical transparent stab with a round weight. If you touch them, it brews really the spores. Actually, Robert Hooke observed the mucor with his first microscopes. Mucor is the first microorganism ever seen by human eyes in details. It's historical mold, as well as it's a social mold. So, September 2013, Chobani withdrew their product from the market. So after more than 300 consumers complained of GI tract discomfort after ingestion of this yogurt, they finally identified mucosal contaminated their entire factory. So that goes to the yogurt. So we were lucky enough to get isolate. So we just analyzed that isolate, the genome and virulence level we published. So as I mentioned, mucor is also social mold. And also mucor is a dimorphic fungus that Louis Pasteur first found in his book, Études sur la bière. So mucor grows as mold with plenty of oxygen. As you can see, spores start to grow isotropically, then germinate. Then this is very typical mold growth. So several decades ago, Bartolomé Garcia rediscovered this mucodimorphism. The dimorphism rely on the relatively high concentration of CO2. If you deprive oxygen and with high concentration of CO2, mucor grow only as yeast. This is multi-budding yeast. So this is completely different pattern of the growth. This depends on the especially environmental cue. Dimorphism is found in many pathogenic fungi. There's a classical group called dimorphic fungi. They grow as mold at 25 degrees, but at the human body temperature 37, they grow as yeast. This is virulent form of them. So Candida albicans can switch their morphology from yeast to hyphae inside the host cell. That is one of their virulence factors. So normally, so Cryptococcus is mainly grow as yeast, but when they mate, they make hyphae. So normally we don't think Aspergillus is dimorphic fungus, but morphogenically different form like Conidia or hyphae both are virulent. So Nature Review paper has a typo here. It should be fumigatus, not umigatus. So we found the mucodimorphism is regulated by calcineurin. So you're going to be hearing more about calcineurin later on. So just briefly refresh your memory. Calcium comes into the cell that binds to calmazulin, then calmazulin binds to the calcineurin complex. Calcineurin has two subunits. Calcineurin catalytic subunit, which has phosphatase activity. Then this is regulatory subunit, which activates calcineurin. So main difference of calcineurin from other phosphatases is the presence of regulatory domain, particularly autoinhibitory domain. So autoinhibitory domain actually blocks the active site. Then calmazulin and regulatory subunit landing there, this active site is exposed to the subunit. So that's why two subunits is required to be active for them. There are two drugs involved in this pathway. So FK506 binds to FKBP12. This complex inhibits calcineurin. And cyclosporin A binds to cyclophilin A. This complex inhibits calcineurin. So there's another drug called rapamycin also binds to FKBP12, and this complex inhibits not calcineurin but Toll pathway. So Joanne Kingsbury from our lab presented a poster about this Toll pathway, so I'm not covering this part. So calcineurin is a good target for antifungal drug. So in other fungal systems, including Candida and Aspergillus, so probably on more fungal systems. As you can see, so Mucor and Rhizopus both are the most responsible mucormycosis fungal agent. So calcineurin inhibitor FK506 dramatically inhibit their growth. You can see the size of the colony in the presence of FK506. Actually, Nina Singh at Pittsburgh found some less incidence of mucormycosis in the organ transplant recipient under FK506 treatment. So one thing you have to make sure is that FK506 also working on human to suppress immunity. So, another study, so they found that FK506 has synergistic effect for Sarcomas in vitro and vivo experiment in animal mucormycosis model. What it found was, so I just mentioned mucor grow as mold. So for different isolate, you see very nice hyper map here. But if you add FK506, they make very compact small colony. If you take a look at them, they completely grow as is, regardless of amount of oxygen around there. This is exactly the same phenotype that you can observe when you grow mucor anaerobically with high CO2 condition. This is first evidence calcineurin is involved in dimorphic switch in this fungus system. We looked at the mucor genome. Mucor, Phycomyces and Rhizopus, they are relatively sibling species. So we found, so mucor genome conserved all calcineurin component. FKB12, cyclophilin A, calcineurin regulatory, catalytic subunit, and chem kinase and calmazulin. I just focus on these two. So interesting thing is mucor has 3 catalytic subunits. This is very unusual. Most fungi have a single catalytic subunit, single regulatory subunit. So one exception is Saccharomyces cerevisiae. It has 2 subunits because of whole genome duplication. But this guy has 3. It's very unusual. So is this really calcineurin? So yeah, we looked at the regulatory domain is conserved, like the drug and immunophilin complex binding domain and calmazulin binding domain and autoinhibitory domain. They are bona fide calcineurin catalytic subunit. So the up there is phosphatase activity domain. So we analyze mutant. Actually, we isolate FK506 resistance mutant. As I mentioned, mucor grow as yeast in the presence of FK506. But we some mutant grow as a mold. How we isolated this, you see the poster of this meeting. Mucor yeast colony in the presence of FK506. As time goes on, you see mold sectors coming out of this. This is we call resistant mutant against the FK506. Some of them, I say some of them, not all of them, some of them carries mutation in the regulatory subunit or catalytic subunit. So that makes sense. Then what mutation does that? We build homology modeling of mucocalcineurin structures based on mammalian calcineurin. This is catalytic subunit. This is regulatory subunit. FKBP12 and FK506. This is actually active site, which is blocked by the drug complex. So substrate don't have access to go to the active site. So this is the way the calcineurin activity is blocked. When those are bubbles indicate the mutation area where we have amino acid substitutions. All mutation reside in the interface area between calcineurin and drug complexes. So presumably the mutation play a role in the interaction between drug and the calcineurin. So for example, CMBR-3 mutation, which alters valine to phenylalanine at 122nd residue. Phenylalanine is larger than valine. So this is valine. Phenylalanine will tilt toward the drug, which results in the closer distance between drug and the calcineurin complex. So may causing the cladic stashes that may be pushing out of the drug or to prevent from the drug coming into the complex. That may be the way this mutant becomes resistant to the FK506. Then we did the critical experiment. So we knocked out the regulatory subunit genes. As I mentioned, if you don't have a regulatory subunit, you don't have a calcineurin activity there. So we knocked out just a single gene that Mucor completely grow as yeast all the time. So we generate yeast-locked mutant by mutating the calcineurin regulatory subunit genes. So FK506 does not have a further impact. That means calcineurin is the only target in the Mucor. So this we genetically prove calcineurin is the key regulator of the morphogenic switch. So genetics and mutation analysis and drug test, everything just conclude the calcineurin orchestrate this morphogenic switch. Then what morphogenics has done for virulence. So we test the virulence in the Gallaria model. Son presented about Gallaria host things, so I don't want to talk about that. So we use Y-type spore, Y-type yeast, and calcineurin yeast-locked mutant and PBS. As you can see, yeast-locked mutant are not really virulent at all compared to Y-type and Y-type yeast. You can make Y-type yeast if we grow them anaerobically. Then the yeast exposed to the oxygen, they become mold. So the inside of the host cell, Y-type exists as mold. So although we just inject spore, they start germinate, grow all the way mold inside of the host cell. But yeast-locked mutant stay as yeast all the time. So it's possible some yeast are less virulent and hyphae are more virulent. Alternative explanation might be analogy like Candida albicans case, the ability of the fungus switching the morphogeny between yeast versus hyphae inside the host may be a virulence factors. Then we looked at how morphogenic difference affected host pathogen interactions. Particularly, we looked at macrophage interaction with the mucospores versus yeast. Spore become hyphae, as I told you. So macrophage phagocytose spore yeast very well. What's going to be happening after that? So this is very typical pathway of pathogen after phagocytosis. This is, after phagocytosis, there's early endosome fusion and late endosome fusion occurs. So through this process, the strong acidification occurs inside the phagosome, activates lots of protease, then start degrading pathogen. This step is called the phagosome maturation. We monitor that step. So cathepsin B is the acidic condition activated protease. So if you have active cathepsin B, that means you have a phagosome maturation occurred inside the macrophage. So we add the fluorochrome dye, which is substrate of cathepsin B. So if this cleaved, then this is fluorescent. It's very simple. If you see red signal, that means it's acidic. So east cell surrounding area, you see red strong signal. It's red halo. That means this is acidic environment. Then phagosome maturation actually occurred with the yeast. But this is not the case with the spores. So phagosome maturation does not occur with the spore. So it seems like mucor spore can escape host defense system, or host does not know they have a mucor inside. So we still don't know what is the real mechanism of this. But by the way, this could be one of the reasons yeast is less virulent than a spore or a mold form. So yeah, this actually we have a mouse mucormycosis model. We infect mouse through the tail vein. Then after day three, we did brain dissections. Inside the brain, you can see hyphal form. Actually, hyphal form is major. They are formed inside host tissue cells. It's interesting, by the way, within 3 days, mucor goes to the brain. So I think we checked the liver cell and other stuff. So very little fungal burden in liver. So major fungal burden only found in brain. So that actually disrupts the mouse's behavior. So mouse seems like just spinning because they don't have a good balance. This mouse spin different axis. So after videotape, we will of course humanely sacrifice them. So we're still curious why they like to go to the brain. So this is, I just the talk was about the morphogenesis and related to the virulence stuff. Then I just get back to the 3 catalytic subunit genes here. I mentioned there are 3. We just examined the expression of them during the morphogenic switch. So we use hyperstage as a control. Then we examine expression of 3 of them during yeast growth by log oxygen or by FK506. We see one of them are highly expressed, the third one, but the other 2 are lower expressed. It seems like they are not completely redundant genes. They may have separate roles. So actually, we have 1 regulatory subunit, 3 catalytic subunit. We successfully knocked out those 2. Then we examined the phenotype of them. So here is the CNA knockout, CNA B knockout. They have a completely separate different phenotype. For example, CNA B is much more sensitive to cyclosporin A compared to CNA A. However, CNA A mutant is much more sensitive to FK506 compared to the CNA B. This is very opposite phenotype, and also CNA A is much more sensitive to SDS. That still says they are separate role in the separate cellular roles. Another interesting phenotype of CNA is their tip splitting phenotype. Mucor hypergrowth is just one single direction. It goes very well. But this mutant, as you can see, at this moment, hypertip is just split. It's a multidirectionally grows. That means this mutant fails to maintain hyperpolarity, but this does not happen with this CNAB mutant. Still says this is a separate role. But seems like Mucor evolved 3 or multiple catalytic subunit to assign kind of separate role during evolutionary trajectory. Other interesting phenotype of CNA is they change the size of the spores. In our previous study, we found spore size is linked to the virulence of the mucor. The larger, the more virulent. It's simple. So what we find found was CNA mutant produce larger spore than wild type. That's pretty larger. That prompted us to test the virulence of this mutant. So here, in the given number of inoculum, wild type is moderately virulent, but these CNA mutants are very aggressive. It kills the host cell pretty quickly. But CNA mutants are as virulent as wild type. They are not more virulent, they are not less virulent. Still, they are different role. But observing this result, this could be paradoxical. Because we just claimed calcineurin as an antifungal target, then we knocked out the calcineurin gene that is more virulent. That is not what we want to see. But our hypothesis is still valid. As I showed, CNA mutant and CNAB mutants, they are much more sensitive to the calcineurin inhibitors. So how this works? We found some synergistic effect of calcineurin inhibitor with other antifungal drugs. In a test with the CMBR, this is regulatory survey mutant. So Envisome actually does not display pretty much different MIC between wild type and the mutant. But mycomin, this is the highest concentration we can achieve with mycomin. So still, mucor grow really fine. But the mutant, you see dramatically reduced MIC of this drug. The same thing, posaconazole, MIC dropped significantly. So in a test with the CNA mutant, so actually Envisome does not change, Envisome MIC is not changed between wild type and versus the mutant, but in the presence of a small little amount of FK506, overall MIC is a step down, going down. The same thing true, mycomin actually does not work at all, but in the presence of a small amount of FK506, the MIC goes down. So same observation is here with the CNAB mutant. So Envisome, in the presence of FK506, MIC goes down. The same thing, in the presence of FK506, MIC goes down. So this is as an in vitro experiment with the analysis of a genetical mutant and also analysis with the drug combination. This says synergistic therapeutic approach could be working. So now we are moving on to test this in the mouse model. So then I'm moving toward the conclusion of my talk. So I mentioned calcineurin is more like virulent hyperform because we inhibit calcineurin by mutation or by drug. We see that they grow as yeast. But one should think about protein kinase A because I mentioned high CO2 plays a major role during the yeast growth. CO2 diffused to the cell and converted to bicarbonate by cytosolic carbonyl anhydrase. We confirmed high concentration of bicarbonate induced the yeast growth. Then literature says cyclic AMP actually control their dimorphic switch. So this all the way says protein kinase A is toward the yeast growth. I mentioned calcineurin is toward the hypergrowth. What's their relationship? Very simple explanation is this is phosphatase, this is kinase. They are doing exactly opposite things. They may share the same substrate. They're fighting each other or competing each other. But we found otherwise, evidence. So we measure the pKa activity in the hyphae cell. So we have some relatively lower activity because I said this signal pathway is not turned on. But if this signal is turned on in anaerobic condition with high CO2, as you expected, you can see higher pKa activity. But if you inhibit calcineurin activity by mutation or by drug, pKa activity still stays high. So somehow, if you diminish calcineurin activity, then pK is more active. It's not like sharing substrate. It's like there's some kind of antagonist relationship, possibly calcineurin negatively regulate pKa. So we do not have any evidence to address that yet, but this is very interesting observation. This pattern is also true in other fungal systems. When we inhibit calcineurin with FK5 or 6, we saw pKa activity goes high in Rhizophus, Cryptococcus, and Aspergillus. So this is not only happening in Mucor, but also in other fungal systems, probably in other eukaryotic systems. So just grand summary, I didn't say this pretty much very well, but briefly mention just larger spore, the more virulent. It's simple because the larger spore can escape, innate host immune system than smaller spore. So calcineurin is in the centers because they control the dimorphic switch. And as I mentioned, one of the calcineurin mutant makes an even larger spore. So calcineurin might be the negative regulator of spore size. Then protein kinase A is here. So one of the calcineurin mutant is more sensitive to SDS that says calcineurin may be involved in cell wall integrity. Then yeast, Staphylococcus spore, or hyphae has different outcome in host pathogen interactions. So as I mentioned, our virulence tract is centered by calcineurin. So there's no question calcineurin is the right choice to treat these fungal infections. So later on, you will hear more from Bill and Mitchell about calcineurin study in entire fungal systems. So our lab's big boss, Joe, and so I thank him, and our immunology partner, Marty and Dennis, and also Lars, Jude, and Aberdeen, and all Heitman lab members. Santiago and Rosa helped us to build the mucogenetics in the lab. Ibrahim helped us to develop the animal model at Duke. And Nina, Francois, D sent a lot of clinical mucor isolate that we have very nice collection of clinical isolate of mucor. The JGI Broad Institute sequenced mucor genomes. So this work is funded by Astellas. They provided FK506, mycamine, and Amazon as well as funding. It's really helpful to conduct this research. And NIH funding, and also I'm an alumnus of the MMPTP program. So with that, I'm more than happy for answering your questions.